So in mammals so far, we've thought about random X inactivation, the fact that you could have either the maternal or paternal X chromosome inactivated within the female nucleus. However, this isn't the only form of X inactivation that occurs. So we know that random X inactivation occurs around gastrulation, the choice is heritable. But a second type of X inactivation can occur that is studied predominantly in mice and also in marsupials, called imprinted X inactivation. In this case, there isn't a random choice about which X chromosome is selected, but rather it's dictated that it will be the paternally derived, the X chromosome derived from the father, that will be chosen to be inactive. So we know that this form of imprinted X inactivation, at least in mice, occurs in pre-implantation development, so that's before the embryo has made a placenta, and then occurs in the placenta itself. It also, it's interesting to note that while it doesn't seem to occur particularly frequently in human cells, in marsupials, it occurs in all tissues. So marsupials, you might know, are non-placental mammals that are indigenous to Australia and South America. And in these marsupials, these non-placental mammals, imprinted X inactivation is occurring in all tissues, so including in the adult marsupial females. And so because of this difference between having it occasionally in mice and having it all the time in marsupials and very rarely or never in adult, in female humans, it's argued that maybe this imprinted form of X inactivation, the case where the choice is removed and it's no longer random, is perhaps an archaic mechanism. So let's think about when each of these types of imprinting occurs in the mouse. And for most of the X inactivation lectures, we're going to be predominantly thinking about what occurs in the mouse, because we can learn a lot about the molecular mechanisms in a model system like the mouse, and these studies are much more challenging in humans. Although some of these studies have now been performed in humans, they've been much more recent, because it's only been very recently we've had the ability to perform these studies in humans. So here I'm showing you a picture of kind of the circle of life, if you like. So we start out and we have a sperm and an oocyte. They fuse to form a zygote and then go through the pre-implantation phase of development, going through from the 2-cell stage, the 4-cell stage, the morula, and ending up at the blastocyst that I mentioned. So somewhere between the 2 and the 4-cell stage, this is when imprinted X inactivation first occurs in the mouse. And so this is where the paternal X chromosome is silenced. So we're not exactly sure, and it probably depends on which locus you look at on the X chromosome, exactly when this first occurs, but it is certainly between this 2 and 4-cell stage. So for this pre-implantation period, it's only the maternally derived X chromosome which is active. So we then form the blastocyst, and the blastocyst is a ball of cells that has on the outside a layer of cells that will go on to form, which is called the trophectoderm, which will go on to form the placenta. And within that ball of cells is the inner cell mass. This inner cell mass here, depicted here, will go on to form the embryo itself, and therefore also go on to form the adult. So these two different parts of the blastocyst are actually treated differently in terms of X inactivation. So the trophectoderm that will go on to form the placenta maintains the imprinted X inactivation that was found in that pre-implantation period. So the X inactivation that was first established between the 2 and 4-cell stage is then continuing to be maintained in these extra embryonic tissues in the placenta that will support the growth of the embryo. However, the inner cell mass of the blastocyst, which will go on to become the embryo, does something different. So a blastocyst is present at around E3.5, so embryonic day 3.5 to embryonic day 4.5 of gestation in the mouse. And at embryonic day 5.5, we know that random X inactivation takes place. This is when you have this gastrular stage of development that's drawn here. At this gastrular stage of development, in the epiblast is the portion of the embryo that will actually go on and make the embryo itself and the adult being in the end. And the rest of it are really membranes that are going to become the placenta. So what we now know is that within the cells of the inner cell mass, these cells will have to remove the imprinted X inactivation, remove that paternal imprint, the silencing of the paternal X chromosome, and then start random X inactivation afresh at embryonic day 5.5. From this point onwards, this random X inactivation is maintained. So it's maintained through to the somatic cells of a mid-gestation embryo shown here, and then maintained through to the somatic cells of an adult female. So seeing as there has to be a switch from imprinted X inactivation to random X inactivation from the inner cell mass to the epiblast, something must be happening there. And indeed, what we actually find is that the cells of the inner cell mass contain 2 active X chromosomes. And this is extremely unusual. It happens just twice in the development of a female mammal. First in the inner cell mass, and this is because they need to clear that imprinted X inactivation that was found earlier in development in that pre-implantation phase. And secondly, it happens again in the primordial germ cells. So the primordial germ cells are during the development of the eggs, and then what will happen, or sperm, but in this case the eggs, you find that X inactivation is again cleared or removed. So here you again find 2 active X chromosomes. So these 2 exceptions to the rule are interestingly both pluripotent cell types, meaning that they are able to give rise to all of the lineages in the embryo. And so there seems to be some link between pluripotency and X inactivation status. In this case, that you don't have X inactivation in these cell types. So what we also know is that cells of the inner cell mass can be used to derive embryonic stem cells. So you may have heard something about embryonic stem cells, but these cells can be used again to derive any of the embryonic lineages, but not the placenta. The placenta, as I mentioned, comes from the trophectoderm. And so the embryonic stem cells can't make the trophectoderm, can't make the placenta. And these embryonic stem cells have been extremely useful for studying X inactivation, because when we can derive embryonic stem cells from the inner cell mass, they can be grown indefinitely in culture. And when we do that, we can therefore have many, many more cells than could be derived from the inner cell mass itself of a small embryo. But additionally, if we take these embryonic stem cells and stimulate them to differentiate, then they will undergo X inactivation, random X inactivation, just as it occurs during normal development from E5.5 onwards. And so much of what we've learnt about X inactivation, at least in the mouse, has come from studies of embryonic stem cells that have been derived and differentiating them in culture. And so over the subsequent lectures, as we go through each of the different mechanisms involved in X inactivation and the stages of X inactivation, much of what we've learnt has been coming from those embryonic stem cells, with a smaller number of studies that are actually done in embryos that are freshly taken.